Welcome to Double Fertilization 101. We're your hosts, Anika,
Jacob.
And Delano.
and we're so excited to introduce you to. Wait, what's that noise?
Hi there, I'm Buzz the Bee, and I'm going to be helping to guide you through a process that us bees are integral to pollination. So, what do you say? Are you ready to dive deep into double fertilization?
Of course, Buzz. Lead the way.
All right, let's go. So, let's start with the basics. Double fertilization is a unique trait of flowering plants.
That's right, Buzz. Flowering plants, or angiosperms, are the only ones with double fertilization, which sets them apart from gymnosperms and other vascular plants. That's not all that makes them different, though, so let's go through the reproductive morphology of angiosperms. Angiosperms have flowers, which are organized into different parts called whorls. The outermost whorl is called the calyx, which includes the sepals. Next, we move on to the corolla, which contains the beautiful petals of the flowers. After that, we come to the andrecium, which holds the stamen composed of the anther and filament. Finally, we have the gynecium, which is comprised of a carpel or carpels, which hold ovary, style, and stigma.
Wow, that's a lot of parts. Us bees have to work through all of the layers to get to that sweet nectar. When we do that, some of the pollen from the anther gets stuck to us, and we can deliver that pollen to the stigma.
Exactly. This pollen develops in the microgametophyte, the male haploid gamete-producing plant phase in the anther. Within the ovary of the plant, you can also find the megagametophyte, which is the female version. The megagametophyte undergoes a two-step developmental process that results in an embryo sac that looks like this. We have the chalazial end, which houses the antipodal cells, and the micropilar end, at which the synergids and egg cell reside. The diploid central cell contains the two polar nuclei, which are vital to the process of double fertilization.
That's right. There's a lot of steps involved in angiosperm fertilization, but it all starts with the pollen in the anther. That's where our game begins.
Hi again. I've got a very busy life, you know. I'm always going around pollinating all the flowers I can see. Every once in a while, I find something unexpected. And guess what? Today, I found a video game.
That's right. Buzz discovered a video game at his local arcade about double fertilization. He and his friend are able to play together as pollen cells to try and fertilize an angiosperm flower. Level 1. The Transport. In this level, Buzz and his friend begin their journey as two haploid pollen cells in a pollen grain. The game starts with a scripted journey where a bee takes this grain from an anther of a flower to a stigma of another flower. Level 2. The Tube. Here, Buzz and his friend must begin their trip to the ovary. To do this, the pollen tube begins growing down the style of the flower from the pollen grain. When it is complete, the two pollen cells are able to travel down into the ovary. Level 3. The Guards. On this level, the pollen cells must try and enter the ovule. The problem is that two synergid cells are blocking the path. This ends up not being that bad of a problem though, as when the pollen cells get closer, the synergid cells begin to disintegrate and allow them into the ovule.
Oh golly, that was a close one. Good thing we got through.
Level 4. The Ovule. Two new tasks have just been assigned to Buzz and his friend, and they must split up. One pollen cell must head towards and reach the haploid egg cell, while the other pollen cell must reach and merge with two polar nuclei. Level 5. The Egg and the Polar Nuclei. Buzz heads towards the egg and then begins fertilizing it. Him doing this has now turned them into a diploid zygote. His friend has an even stranger path though. When he merges with the polar nuclei, the three cells merge together and form a triploid endosperm. Level 6. The Fruit. This is actually the end of the video game, but the story isn't over. The ovary will grow and swell, eventually forming a fruit. The zygote continues to grow into an embryo and then eventually into a seed. And the endosperm will grow and become a storage place for nutrients to help the fruit develop. The fruit and seeds are now free to be dispersed and eventually develop into a new plant, and this whole process will then repeat in the new flower.
Okay, now that we have an overview for how double fertilization works, let's zoom in a bit on what's happening. If you remember, the pollen tube grows toward the synergid cells in the ovary. But this begs the question, how does the pollen tube know how to get there? Well, the synergid cells secrete lure peptides, which attract the pollen tube and guide it to the ovary. Much of the pollen's navigation happens in the transmitting tract, a stretch of nutritive tissue within the style of the flower that facilitates the pollen tube's travel. See, Buzz? This is how you were able to get through in the pollen granule game.
Wow, cool!
For this next part, we'll check out my rendition in MS Paint. Once the pollen tube reaches the synergid cell dies and disintegrates. We still don't fully understand the signaling happening in the synergid cells, but it is hypothesized that V-ATPases and adapter protein 1, AP1G, play a role in degrading these cells. Interestingly, it has also been suggested that these signaling molecules may prevent multiple pollen tubes from growing to the same synergid cell. After the first synergid dies, the other undergoes apoptosis and dies as well. Buzz, you look like you have a question.
Yeah. What do those antipodal cells do?
That's a good question, Buzz. We don't actually know if those cells have a function. All we know is that they are present in the ovary, and they persist for some time after fertilization. Mysterious, huh? As we talked about in the overview, double fertilization will now occur. One pollen joins with the egg cell, and one pollen joins with the two polar bodies. This gives us a zygote and triploid endosperm, plus antipodal cells. Oh, Buzz, another question.
Yeah. Why is it that the endosperm is triploid? I mean, gymnosperms seem to do just fine with haploid endosperm. Why would a complicated process like this need to evolve in angiosperms?
That's another good question, Buzz. The explanation for the triploid endosperm has a lot to do with imprinting, a genetic process where an allele from one parent is excluded from being expressed. Triploid endosperm has been shown to be selectively favored due to the fact that there are two copies of the maternal chromosome in the endosperm, making the maternal endosperm phenotype more expressed. Basically, the trait is favored by selection because, in the case of imprinting, it causes other competing genes to be excluded more frequently, which increases the fitness of the maternal chromosome that has triploid endosperm. So, in conclusion, double fertilization is a carefully controlled process that allows plants to produce seeds with triploid endosperm, a trait maintained by chromosomal selection.